Okay, it is a couple of minutes after noon, and I'm going to go ahead and get started with today's presentation, which is fairly brief. I may pause from time to time and check to see if there are people waiting to be let in. So please be patient with us if that is going on. Let's see, here's another couple of folks joining now. All right. So I would like to begin by welcoming everybody who's joining us today for our annual update call. My name is Sharon Presnell, and I serve as the president here at the Amnion Foundation. And whether you are already familiar with the foundation or you're joining us today to learn a little bit more about our mission, I hope that you will find today's session informative, and please do submit any questions that you have as I go along in the chat box that is a component of the Zoom window. And as we go along, Anna Smith, who's our head of operations here at Amnion, will ensure that those questions get asked at the end. And then you can unmute at the end also and ask questions if you'd like. And so I am going to turn off my video for the remainder of the presentation, just so that we don't run into bandwidth issues, which is a typical kind of problem on Zoom. And so just be patient with me for a moment while I do that. Okay, and I'm going to try to keep an eye on this participant window if people come in as well. So today's presentation is intended to be a very high-level status update from the Amnion Foundation that will provide a brief history for newcomers, but also cover the challenges and accomplishments that we have in 2020. We'll also share our 2021 goals and discuss the importance of the community engagement in the future of the foundation. So moving on here, I want to talk a little bit about the history of the foundation. So Amnion was founded as a not-for-profit. We're a registered 501c3 organization with a mission to collect and bank unique stem cells that are found in postpartum birth tissue, specifically the placenta, to support regenerative medicine therapies. These placental stem cells were originally discovered by Dr. Anthony Atala, who now leads the Wake Forest Institute of Regenerative Medicine. The cells are very unique in that they are multipotent, meaning they can generate multiple cell types in the body. They also have robust immunomodulatory properties and are under active investigation preclinically and clinically for treatment of conditions such as Crohn's disease, osteoarthritis, and wound healing. So there was a private donor gift that came into us from the community, and this enabled labs to be built. Around 2016, this was completed, and we are located in Winston-Salem. And by late 2018, all the work had been completed, meaning we had facilities, we had hired staff were trained and competent to enable the foundation to produce GMP, good manufacturing practice, compliant placental cells for clinical use. And that was a really fantastic accomplishment. The demand, though, for clinical grade cells at that time was just not high enough to sustain us fiscally continuously banking GMP grade cells at the foundation. So in 2019, we expanded our mission to support more broadly the use of these placental stem cells, as well as other birth tissue associated cells in the development of new pharmaceutical and regenerative medicine therapies for underserved diseases. So this meant a couple things for us that were really important. One is that we would need to broaden the repertoire of cells that we generate to extend beyond those placental stem cells. We're still making those, but just to include all of the relevant cell types that are found within the placenta and umbilical cord so that we can better serve the advancement of new treatments and cures. And the second thing is that while we would still retain our capabilities for clinical cell production and banking, this would not be a primary short-term focus or a continuous activity because of the expense associated with that. And so moving on from there, let me see if I can get the slide to advance here. So just a word about how does the Amnion Foundation, with this amended mission, how do we fit into the big picture of regenerative medicine and drug development? So with the expansion of our mission to include the broader support of earlier stage preclinical research programs, we now offer partners a seamless solution from that first experiment all the way through production that they might require, whether those are regulated preclinical studies for drug development submissions or whether they're clinical studies for regenerative medicine therapies. It's helpful, like the diagram on the left, to just think of the Amnion Foundation and the cells and services that we provide as a platform or hub that serves regenerative medicine and drug development pretty broadly. Research programs ultimately translate into new pharmaceuticals or cell-based treatments. And by focusing on what it is that we do best, which is generating quality, well-characterized cells from birth tissue, we are partnering in the advancement of many novel therapies for underserved diseases. And this is distinct from, say, placing all of our bets on a single indication and supporting that indication, from soup to nuts. So our goal is to operate sustainably in this and support as many therapeutic advancements as possible that are relevant to fetal maternal health or regenerative medicine applications. And, this is really important to us at the foundation, that our mission is really not around maximizing the profit that we get per unit of cells that are provided out into the community, but rather that we are operating sustainably in supporting these advancements. And this is meaningful to us at the foundation, and I think it is also very meaningful to people who donate to us, both tissue and finances. And it's important to the partners that we work with that they know they've got someone focused on the quality and making things happen versus how many profits are generated. And so our mission today, just to state that really clearly, is to ensure that the valuable cells found within the birth tissue are made broadly accessible to support groundbreaking research and to support the development of life-changing therapies. We are doing our best to make the most of every tissue that is donated and provided to us by isolating not just those placental stem cells that we were talking about earlier, but the full spectrum of cell types that can be used in the development of therapies. Our current capabilities allow us to provide custom cells and services for our partners from research all the way through their GLP or GMP compliant production. It's also really important to realize we do work with birth tissue at the Amnion Foundation, and there have been at times, I think, some controversy about that out in the field. And it's important to understand that we work solely with full-term placenta and umbilical cord that is collected locally here in our community under informed consent and only after the birth of the baby. And this tissue would otherwise be discarded. And the collection poses absolutely no risk to the mother or the child. And it allows our financial supporters and our partners to be comfortable sourcing materials or supporting that sourcing through the Amnion Foundation because there simply are no ethical concerns. And so I think with that, you hopefully have an understanding of what our mission is and what we're trying to accomplish with the donated tissues that we receive. I'd like to move now into speaking a little bit about what we were able to achieve in 2020. But first, I want to cover some of the challenges that we had for 2020. So I'm sure you're all very well aware as we sit here on this Zoom conference instead of being in person ever. is that in early 2020, our world was really transformed as the result of COVID-19. And we made a lot of necessary operational changes to ensure the safety of our staff, the clients that we work with, and partners, while trying to continue on our mission throughout the year. The operational impact to us was that we were really able to process a lot fewer tissues than we would have normally liked to process because of those operational delays and also financial challenges. The economic impact to us, many of our partners and clients had delayed, reduced, or even ceased operations. This disrupted our revenue streams because we do, not-for-profit organizations do take in revenue to do the work that we do. And so that supports us in addition to the financial gifts that we receive from our community. So we have shifted then. So moving on from the COVID challenges, so here we are amidst that still, but we also shifted our mission to include the isolation and provision of all these other cell types. And so each one of those cell types had to be developed internally here at the foundation so that we had robust methods to be able to produce those and provide those out to the community. And we were going to need at least 6 additional cell types to really make the most out of each of those placentas that we receive. And several of the cell types that we work with have to be isolated within just a couple of hours of birth, or you cannot obtain viable cells. And so we had to solve some operational challenges there, and I'll talk about that a little bit more on the next slide, how we got there. And then also, we just at the time that we were beginning to offer these cells and services out into the community, every scientific meeting, trade show, and other form of exchange was canceled because of the virus. And so we would have to find then new ways to create awareness of what it is that we offer and the unique tools that we provide at a very, very modest cost out into the community. So talking now about our accomplishments for 2020 amidst all these challenges. So we did solidify a partnership with a local firm, Birth Tissue Recovery, and they are amazing. They've worked with us very closely to make sure that the consent forms that are signed by the donors are extremely robust in support of the projects that we are enabling out there in the community. And they're able to provide us access to these birth tissues fully consented almost immediately after birth. We're getting them within a few hours. And that means that we can do that development work. And in this year alone, this very small team has actually developed the methodology to allow 6 additional cell types to be isolated from full-term placenta. And this includes the isolation of some cells from frozen banked whole tissue. And I can't stress enough how important this is, because sometimes it is a lot more economically feasible to put whole tissue away and then come back to it later as you need it, especially for regenerative medicine applications. We have now available to our partners large vessel endothelial cells. So these are vascular cells that line the umbilical cord. We have small vessel endothelial cells. These are the tiny little blood vessel cells that form capillaries in the placenta. We have very specialized cells called cytotrophoblasts, and these cells are very important because they form the barrier between the mother's bloodstream and the baby circulation. And so they control what crosses from the mom to the baby. We also make mesenchymal stem and stromal cells from the umbilical cord. We are working with placental macrophages, which are fetal in origin, and they are what is protecting the developing baby from pathogens like viruses and bacteria that may cross over from the mother. And then we're working with fibroblasts, which are support cells within the connective tissue of the placenta. We've also established multiple of these cell types at what we call physiologically relevant oxygen conditions. And these pro cells, as we call them, they exhibit a lot more function than cells that are isolated under standard conditions. And in some cases, using these conditions are enabling us to isolate cells and even keep them in culture that you would not be able to do otherwise. And so that's been a pretty big achievement and something that we will be publishing later in 2021. And I just want to highlight a little bit of these because it's such nice imagery. So on this next slide here, if we look at the image on the left, these are some of these Amnion Foundation endothelial cells, and they're shown here in the process of tube formation. And this is a hallmark feature of this cell type. And these particular endothelial cells came from the lining of the umbilical cord. And what they're doing here is making tiny little networks of blood vessels in the laboratory. So very, very important for the study of lots of different diseases, or you could imagine in the treatment of degenerative diseases where you're trying to restore vascularization to an organ that has been damaged. And then if you look at the image on the right, these are those cytotrophoblasts that we were talking about earlier on the previous slide. And what's really cool about what this lab has been able to achieve is that up until now, you could either isolate trophoblasts from a full-term placenta, but you could not grow them any further, or you could use preterm placenta and you could grow the cells up, but you're working with fetal tissue essentially, which is very controversial. So we have been able, and this is very important to the field, we have been able to isolate and grow these cells up and bank them from a full-term placenta. So no controversy, but bringing out to the community a cell type that has not been available before. And for studies of maternal fetal barrier and drug development, this is really important resource to have available for people. So if we continue now on our 2020 accomplishments, I would just like to start by saying, you know, the first step in getting to any new therapy, any new treatment, or any cure is to really conduct that research that is required to test the idea or compare multiple ideas or optimize and refine a potential solution so that it has a better chance of being safe and working when it's finally given to humans for the first time. And so for the Amnion Foundation to achieve our long-term goal of supplying cells to support cures, we have to be there also in the beginning to support the research programs that are going to turn into those cures down the road. And so our short-term goal is to get our cells into the hands of thought-leading researchers that are doing important work in fetal maternal health and in regenerative medicine. So in 2020, we created our SEED grant program, and this allows selected investigators to gain a limited but no-cost access to the Amnion Foundation cells and services that will uniquely enable their programs. This supports our mission by getting our enabling technologies into the hands of leading researchers, and we can continue to partner with them through development of new therapies. It also creates awareness of our products and services through their networks out into the research community. We received our first round of applicants this fall and have selected 3 is what we determined we could support at this time. And they were selected based on a few criteria. The potential impact of their research program. And this is both regionally, things that might be important in our region of the world where these tissues are collected and where many of our financial donors reside, and things that are also very important to global health. We are looking for investigators that have a track record of success in the area of research that they're proposing, so that if we're placing this bet, providing these materials, there's a high likelihood that it's going to be a fruitful effort for everyone involved. And then we also looked at it from our ability and our current status to be able to supply the cells that they need in the volumes that they need to be successful with their programs. And so those were the criteria. And I'd like to introduce you now to our first 3 recipient labs. And we're extremely pleased with the work that is going on in all of these labs, and we hope to be able to support more of these in the future. So Dr. Minjae Song is part of the Early Translational Lab at the National Center for Advancing Translational Sciences, NCATS. This is the newest branch of the NIH. The goal of his work is to create a human cellular model of the maternal fetal barrier that can be used in the drug development and disease modeling arena. Their approach here is to use advanced 3D bioprinting and biomanufacturing capabilities that existed in cats to create precise multicellular 3D tissue models of the maternal fetal barrier. And it's important to note this lab has already been wildly successful at building models like this for human skin, human retina, and other human tissues, and they're very well published in this regard and very well regarded. So the cell types that we're going to be supplying to them include the cytotrophoblast, endothelial cells, and macrophages. And we're really excited to be supporting this lab. And there's a couple of links here that you can check out if you want to learn more about these labs. As I go through them, there's a link for each one of them. So Dr. Shai Soker at the Wake Forest Organoid Research Center, this is part of Wake Forest Institute of Regenerative Medicine. His lab has received a seed grant to create living human cancer models for use in the selection of patient-tailored treatment regimens. And the approach here is to use cells that are obtained through the Amnion Foundation to create living stromal tissue. So this would be the vascular cells, also vascular cells and the fibroblast and other support cells that will be able to then host patient-specific tumor cells in the lab and enable that tissue as a whole to be exposed to different treatment regimens so that things like cancer cell invasion and metastasis can be studied on a patient-specific level. And this will ultimately, hopefully, translate into being able to take more of a personalized medicine approach for cancer. And so lastly, there is a seed grant being awarded to a doctoral candidate at the University of North Carolina in Chapel Hill at the Gilling School of Public Health, Anastasia Friedman, and her mentor, Professor Rebecca Fry, who is a very well-known toxicologist. And these individuals are looking to establish the mechanism of action of some environmental toxicants that are actually really important in North Carolina and known to have some adverse effects on fetal development. They're going to be using the trophoblasts that we spoke about earlier and endothelial cells to look at what happens to these particular cells when you expose them to these known toxicants like inorganic arsenic and PFAs. And these are prevalent in our environment in part due to natural processes and also some industrial processes that are fairly unique to North Carolina. And they're going to be looking at this, but they're also going to take a really cool approach and look to see how the placental stem cells that we kind of began our conversation with today might protect these other cell types from the damage that occurs when you have exposure to toxicants like this. So each of these projects is very, very different and potentially very impactful to where we are headed kind of in that mission of supporting science broadly to advance new treatments and cures. And so talking now a little bit about our 2021 goals, we certainly intend to continue to support our initial seed grant recipients with Amnion Foundation sales and services to enable their projects. We would certainly like to expand this program both to support additional translational projects, but also potentially to support some of these projects at a higher level. We talked a little bit about publishing work from the Amnion Foundation that was completed in 2020, demonstrating some of the functional benefits of establishing cells and maintaining them at physiologically relevant oxygen versus standard lab conditions. And publications and things like this help us get recognized as a supplier out into the community. And this is important to our last goal, which is listed here, that we really need to get to a point of reaching fiscal sustainability by providing our cells and services out to clients, but also increasing our community engagement and support through tax-deductible donations. And that's always been kind of the dual road that we are on. It's becoming more and more important in this economy. And I want to talk a little bit about what a gift to the Amnion Foundation can enable here. I think it's important to know that every dollar we receive is used for purpose. It's valuable to us, and we appreciate gifts of any size. but I want people to have a basic understanding of what dollar values accomplish for us internally and how meaningful they can be. So, for example, a donation of $500 can enable us to accept a donated birth tissue for processing. A donation of $1,000 enables us to bank some whole tissue for future cell isolations. A donation of $2,500 turns a donated tissue into multiple cell types that can be used to support projects for one of our seed grant recipients that you just heard about. And donations of $10,000 or greater actually provide something pretty fabulous, which is partial support for this GMP compliant processing of tissues that can then later be used in the clinic. And we really do welcome discussion with members of our community that may be interested in supporting something specific, and they're looking for an avenue to do that. I think we are particularly well positioned to find out in our community researchers that are advancing new ideas and novel therapies that will align with a particular donor's interest, and we can help facilitate those engagements as well as supply the raw materials that are required to actually make them come to life. And tomorrow being Giving Tuesday, I hope that folks will consider passing our information along or themselves providing us with some resources to help us stay afloat and get to that sustainability goal in 2021. Please feel free to reach out to me directly as well. It's Sharon@amnionfoundation.org, and I'm happy to speak with you one-on-one, answer any questions you might have about our mission and how you might become involved if you'd like. I really like to thank everybody for joining today. We're going to have a little time for questions. And I also want to recognize the donors that have so generously contributed thus far to our mission. And we have really appreciated every dollar and welcome when we are able to host tours and host visitors again. We would really welcome any of you here to participate in those things. And so with that, I will turn see if I can turn my video back on here and entertain any questions that may have come over the transom while I was talking. All right, do we have any questions? You may have to unmute, or if you did put your questions in the chat window.
Hey Sharon, it's Anna. I was wondering if you could maybe speak a little bit about, what translational sciences are. I'm not sure that our community may be exactly sure what translational sciences mean and why it's important. And maybe while you're talking about that, you could also talk about why GMP and GLP and the provision of those cells is important to these communities.
Yes, I'm happy to talk a little bit more about that. So when we speak about translational sciences, we're really talking about, because you can go into the lab and do all kinds of experiments. You can ask any question you'd like, really. But translational science is really focused on, it's a much more surgical approach to asking the questions that are absolute requirements to move from the idea into the preclinical studies into the clinical studies, and then into commercial production of something that is going to save lives and be game-changing. And so when we talk about translational, we're talking about things that are very, very focused on making an end product, whether that's a product for clinical trials or something out in the commercial realm, that is going to be made available for either a therapy or in the pursuit of a drug that's being developed for a therapy. So that was the first part of your, that's the first part of your question. And then as you get into that, one of the things that's very unique at the Amnion Foundation is we can certainly support research, but we can support the more highly documented production, the more regimented production of cells that are required for, say, studies that are going to be submitted to the FDA for drug development. So GLP studies, or GMP, good manufacturing practice, is what that stands for. And so if you're going to take birth tissue that comes in the door and process that into cells, and those cells are ultimately going to be implanted into a person, then you have to treat that completely differently. So that's your GMP production mode. And there's a lot of documentation and qualification and quality checks around that, so that you are, when those cells go into the human, you're doing no harm, and you know everything that you can know about the cells as they go in. Hopefully that's helpful.
Awesome, thanks. What, in your opinion, or in your knowledge, are the most advanced clinical programs today involving cells from birth tissue, involving quote-unquote stem cells?
So there are a few things out there worth mentioning, I think, now, especially for this particular audience. So there's some fairly well advanced studies going on using the cells very similar to the placental stem cells that we talked about in the beginning of the talk for diabetic peripheral neuropathy. So that's pretty far advanced. That's in clinical trials now. There are phase 1 trials for rheumatoid arthritis. You may recall that I mentioned in the beginning that the cells have very robust anti-inflammatory immunomodulatory properties. So that's being leveraged for things like rheumatoid arthritis, which there are some phase 1 trials underway for that. And Crohn's disease, there's trials up to phase 2 already in play for Crohn's disease as well, also leveraging those immunomodulatory properties of the cells. There are also ongoing clinical studies for cartilage replacement. And there was even a trial approved in 2020. If you recall, with COVID, the so-called cytokine storm. So for the very seriously ill COVID patients, there was a trial approved and is still being conducted, testing the ability of those cells to kind of tamp or lessen that cytokine storm in the severely affected patients. So there's a lot going on out there. You can do some searches under clinicaltrials.gov and come up with a lot of things. Wound healing secondary to complications from diabetes, that's another area of investigation. So it's a pretty, one of the cool things about these cells is that they can be applied so broadly. Any other questions out from our community?
Yes, this is Randy Perkins. I have a question. Have you been able to sustain your staff during the COVID crisis?
Yeah, so we have remained operational, and we are all still here. We've changed our practices in terms of how close we get and the PPE that we wear on a general basis versus, when we're in the lab doing isolations, we're all covered up anyway. But we have had some impact fiscally. I think we've all kind of voluntarily taken some reductions to make things work. And that's, I think, where we all want to be with it. That's it's very intentional. We are also very fortunate in that we did apply for and receive some of the PPP funds that were made available, and that has helped us tremendously. And we still have gotten support from donors throughout the year, which has been tremendous. And so I would say we're cautiously optimistic, but we also need to find some avenues in 2021 for things to shift for us. And hopefully that will begin to happen as the vaccines become available and we can get back out there again and do our normal thing, be able to go to trade shows and conferences. But we're intact today, slightly reduced schedule, slightly reduced, funding for everything, but we're trying really hard to keep the mission alive.
Thank you. I appreciate that.
Hi, Sharon, it's Andrea Alamani, and I just had a question about storing those cells. You mentioned something about the oxygen requirement for cells. How does that complicate your harvesting of cells and then storing them? What does that look like in the lab?
Yeah, so that's a great question. Everything that we store, and this is actually a really important point. You know, there are things that we do to make the downstream operations very smooth for everyone. So when someone needs cells from the Amnion Foundation, we're going to the freezer and pulling them out of a bank, and they've already been very well characterized and ensured to be safe. So we're not taking a tissue and making something fresh and giving it to the client and hoping for the best because we haven't really had time to characterize it. So everything that we work with is banked under liquid nitrogen in a big freezer, and it's extremely tested before it's ever given to anyone, which is great. The second part of your question, which is, you know, the oxygen levels and how that impacts things. What we have learned in 2020, one of our most important learnings is that some of the cell types that we need to have access to, in fact, two of our grant recipients would not have the cells to be able to support their projects if we had not been able to figure out that the oxygen requirements for that particular cell type were very different than lab standard conditions. And so, you know, we try to, as soon as we get cells in the door, run those tests and figure out what their requirements are so that we can get them to the point of being in the freezer. And then once they're in the freezer, they're incredibly stable. And at that point, we can give our clients really great directions on how to grow them and keep them happy once you get them back out in culture.
That's great. Thank you.
Any other questions out there?
Sharon, I was wondering if you could talk a little bit more about how the foundation works with financial supporters to support their specific interests. So if they have a particular disease or, you know, particular region of interest, how the foundation works to support those goals.
Okay, I can do that for sure. So, you know, one of the things that we have working in our favor here is that we're very small. We are a private operating foundation, which means, you know, we do stuff versus just handing out funds. And we're very agile. So, you know, under that umbrella of we're really focused on maternal fetal health, and we're really focused on, you know, the stem cells and other cells that come out of birth tissue and how those might impact a regenerative medicine therapy. So we do have kind of an umbrella that we operate under. But let's say somebody had a great interest in figuring out if there were novel approaches or novel therapies for diabetes, or for COVID-19, or for you know, Crohn's disease that might involve the cells that we make, that could potentially involve the cells that we make. We are sitting in a really unique position to be able to work with that donor, find out what is really feasible with the dollars that they have available, and then actually find the third partner, which is the researcher in the lab or the clinician who is developing that therapy. And then we become the enabling connector between that donor and the lab who's going to advance that therapy. And we would be then the provider of those cellular materials that would enable both parties to achieve what they wanted. And so we can offer a tremendous amount of customization on that front, and how much involvement would they like to have. And I think that amount of flexibility is pretty unique. And also just being right here in North Carolina, there are a lot of things going on. As you see, even two of our Sea Grant recipients are from universities here in North Carolina. And one of those labs is studying a problem that is very specific to North Carolina. And so there are lots of opportunities for someone's passion to really be addressed with us in a custom way by finding the right lab, finding the right cells, putting these things together. Again, with a focus on achieving the mission versus a focus on having a profit. And I think that's a great opportunity for someone who might want to be a donor.
Great, thanks so much. And I guess, you know, the last question I would like to ask you as the head of operations, what cell type from these birth tissues that we developed in 2020 proved to be the most challenging. I know which one I would pick, so I was wondering if you would pick the same one.
Oh, I think they've all had their special moments. Certainly the trophoblasts, the cytotrophoblasts were a handful, and I think that's been a very tricky cell for us to work with, and I am sometimes amazed at what we've been able to achieve on that front in such a short period of time. I also think the small diameter blood vessel endothelial cells have been quite challenging, and you've been working with those a good bit. But the upside of that is I think we've got a couple of cells to offer out into the scientific community. They're going to be very, very unique in just their existence, but then also their ability to address specific problems and challenges out there. Well, I think we are towards the end of our time today. In fact, I think we're over. We are going to be doing, beginning in January, we're going to have a regular webinar series, and those will sometimes be high-level updates like these, and I want to try to do one of those once a quarter. But then in between that, we're going to have some technical presentations. If you desire to get deeper into the science, we're going to be having some more technical scientific presentations about the things that are going on out there with birth tissue related cells and some of the advancements, and that's exciting, including having our seed grant recipients come and talk to us specifically about the work that they're doing and how that's advancing. And so we're super excited to have that work take off. We are in the stages now of actually providing out the cells to the folks, and that's probably also a speed record. I think we got applicants in September, we made selections in October, and now cells are going out to those labs. So you can't beat that for service. So hopefully that will continue to bear fruit for us. And again, I really appreciate everyone's time today and the thoughtful questions. And if you have any other questions, don't be shy about emailing at info@amnionfoundation.org. Or again, you can email me directly at Sharon@amnionfoundation.org, and we're happy to chat with you. All right. Oh yes, and Anna reminds me, we do have a YouTube channel, and you can watch some additional technical presentations and videos there. And you can find us on LinkedIn and Facebook as well. And with that, I will sign off, and I really appreciate everyone's time today.
Thank you, Sharon.
Thank you. That was fantastic.